A new genetic tool maps how cells choose which of their 20,000 genes to activate at any moment. This is Planetterrian Daily, episode 138. EMBL Heidelberg researchers created a screening approach that tests thousands of regulatory DNA sequences in parallel inside living cells. The assay measures how each sequence influences nearby gene output under different conditions. It distinguishes between sequences that act as switches versus those that fine-tune expression levels. Results showed that many regulatory elements function only in specific cell types or metabolic states. The team plans to apply the method to disease-associated variants whose effects remain unclear. Output can shift tenfold with changes in cellular conditions. Now, here's something most people get wrong about how genes work. Most people picture the genome as a fixed instruction book that is read the same way in every cell. In reality, each cell constantly edits which pages are open. Right now, as you listen, transcription factors and chromatin remodelers are sliding along your DNA, exposing or hiding promoters so that only a few thousand of your roughly 20,000 genes produce RNA at any instant. The new screening method from EMBL Heidelberg shows that many regulatory sequences act only in particular cell types or nutrient conditions, not as universal on switches. One memorable detail is that a single regulatory element can change output tenfold or more when the cellular environment shifts, illustrating how flexible the same DNA can be. The practical takeaway is that future therapies aiming at gene regulation will need to account for cell state context rather than assuming a gene is simply on or off everywhere. Watch for follow-up studies that test whether disease-linked variants act through these context-dependent switches. Researchers at the University of Tartu identified a previously unknown transporter that pumps fluoride out of cells when primary defenses are overwhelmed. The system activates at concentrations that kill most microbes yet leaves the bacteria viable. Because fluoride is used in industrial processes, the finding may help engineer strains for greener chemical synthesis that tolerate higher fluoride loads. Genetic knockouts confirm the transporter's necessity under stress. The work also suggests why some environmental bacteria persist in fluoride-rich soils. From microbial defenses, we move to a method that could help trace the origin of another natural resource. Ratios of oxygen, carbon, nitrogen, and strontium isotopes vary systematically across Amazonian tree species and regions. Researchers built a reference map from verified wood samples and showed that seized timber can be matched to within a few hundred kilometers of its origin. The method requires only a small core from each log and works on processed boards. It offers enforcement agencies a forensic tool independent of shipping documents. Field validation continues with additional species and soil types. University of Maryland scientists isolated endogenous inhibitors that rattlesnakes use to neutralize their own venom. They combined these proteins into a laboratory mixture that blocked venom from multiple viper species at 1/10 the dose of current antivenoms. The product requires no animal immunization, lowering cost and batch variability. Neutralization occurred across geographically distinct venoms in mouse lethality assays. Scale-up and regulatory toxicology studies are the next milestones. The rattlesnake-derived antivenom uses proteins the snakes themselves evolved to block their venom. Because the components are already compatible with viper venoms, the mixture neutralizes toxins from several species at far lower doses than horse serum products. Production avoids animal immunization, which removes both cost and the risk of serum sickness. Researchers identified a chemical approach that arrests tooth decay without drilling or filling. The treatment targets early lesions by promoting mineral redeposition directly into enamel. It avoids mechanical removal of tissue and preserves natural tooth structure. The work focuses on non-invasive intervention at the stage before cavitation occurs. Next steps include larger human trials to confirm durability under normal chewing forces. Men who score higher on measures of desire for social dominance endorse more rigid appearance norms for women. the association held after controlling for age, education, and political orientation. Researchers interpret the pattern as one route through which gender hierarchies are maintained. Data came from validated psychological scales administered to several hundred participants. The study adds evidence that beauty expectations can serve status maintenance functions. Flavobacterium species, previously known only from Asia and the United States, were isolated from farmed fish in Brazil for the first time. The pathogens cause columnaris disease, which produces skin lesions and high mortality in tilapia and other species raised for food. Researchers confirmed identity through molecular sequencing of isolates from multiple outbreaks. The finding raises questions about how the bacteria crossed continents and whether existing vaccines cover the new strains. A refined pulse sequence reduces the magnetic field strength needed to flip bits in certain memory materials by several orders of magnitude. The approach exploits resonant coupling between the pulse waveform and material properties rather than brute force reversal. Lower energy per switch matters for data centers whose power draw continues to rise with AI workloads. Prototype devices retain data stability after repeated cycling. Further materials engineering is required before commercial integration. Lucil tRNA synthetase helps tumor cells maintain protein synthesis when nutrients or oxygen are limited. Blocking the enzyme in mouse models slowed growth of established liver tumors without affecting surrounding tissue. The mechanism involves Lars sensing amino acid availability and adjusting translation rates accordingly. Human tumor samples showed elevated Lars expression correlated with poorer outcomes. Researchers are testing whether existing Lars inhibitors can be repurposed for oncology. A synthetic RNA therapeutic binds conserved sequences shared across several RNA virus groups. In cell culture, it reduced replication of both influenza and coronaviruses without triggering strong interferon responses. The design uses chemical modifications that extend half-life in serum while preserving target recognition. Animal testing is planned to measure protection against live virus challenge. The approach aims to address the mutation problem that defeats many sequence-specific antivirals. In a cohort of 935 older Mexican Americans, individuals reporting both chronic pain and depressive symptoms scored roughly 4 points lower on a 30-point cognitive battery than those with pain alone. The difference appeared at baseline and remained stable over 6 years of follow-up. Rate of decline did not differ between groups. Researchers controlled for education, diabetes, and hypertension. The pattern suggests additive rather than synergistic effects on current function. Longer studies are needed to detect any late divergence in trajectories. While that work improves an existing medical tool, another team has built an entirely new instrument for watching cells in action. Engineers built an instrument that captures large fields of view while maintaining resolution sufficient for live cell tracking. The design uses parallelized optics and fast scanning to record thousands of cells simultaneously over extended periods. It targets samples that change shape or position rapidly, such as developing embryos or migrating cell sheets. Early tests demonstrated stable focus across multi-hour acquisitions without phototoxicity spikes. The platform is intended for labs needing population-level statistics rather than single-cell detail. Tools that let us observe living systems at scale also help us read the history preserved in ancient ones. Microfossils from Precambrian rocks show intertwined filaments and algal cells in arrangements resembling modern lichens. The structures predate the earliest undisputed land plants by roughly 200 million years. If confirmed as symbiotic, the find pushes back the timeline for fungal-algal cooperation that later enabled terrestrial colonization. Thin section imaging and chemical mapping distinguish the two organism types within the same specimen. Additional sites are now being examined for comparable associations. A 100-million-year-old beetle preserved in amber displays antennal and abdominal structures used today for light-based communication. Micro-CT scans revealed sensory pits and light organ precursors absent in non-luminous relatives. The specimen belongs to the same superfamily as modern fireflies, indicating these traits evolved well before the Cretaceous-Paleogene boundary. Comparison with extant species suggests gradual elaboration of the signaling system rather than sudden emergence. Additional amber deposits may yield transitional forms. Researchers at UAB and CRM developed equations describing how protective coatings around phages behave inside the stomach and intestines. The model predicts release timing and survival fraction under varying pH and enzyme conditions. It identifies coating thicknesses that preserve enough viable phage to reach target bacteria without excessive delay. Laboratory validation used simulated gut fluids and mouse dosing. The framework helps optimize phage therapy formulations for oral delivery. Precision mapping of origins also appears in efforts to deliver living therapeutics intact. Next time, we'll be watching for further validation of the isotope mapping method on additional timber species. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at Nera Network. Links in the show notes. And before you go, this show is part of the Nera Network, a family of daily podcasts on tech, science, and markets. Each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Models and Agents for Beginners. Artificial Intelligence Explained Simply for Beginners and Curious Teens. Find every show free at neranetwork.com. The age of AI puts real builders on the phone with an AI host. Apply at neranetwork.com. Age of AI apply. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.